The next speaker is Dr. Daniel Lee, and he's talking about preclinical research, and he's from Florida. He's a professor at the University of South Florida at Tampa. Thank you.
Well, first of all, thank you, Robin, for the invitation. Thank you all for coming out today. I'd like to share a little bit of research about what I do in my lab, give you a little bit of information about what polyamines are. Again, that's my facility. So we know what tau is. Tau is normal, has a normal function. It stabilizes these structures called microtubules, and that basically supports neuronal function. In our tauopathies, again, as you know, tau misfolds. It can fall off the microtubules and it starts to clump or aggregate. Again, as far as there's a number of tauopathies that have already been identified, PSP, cortical-basal degeneration. And so again, it forms these structures or tangles that are hard to digest for the cell. So what are we doing as far as different approaches? Again, researchers are literally trying to attack this from all different angles. Some of these include trying to stabilize the microtubules, as Dr. Boxer mentioned. Some of them involve trying to decrease the clumping and the aggregation. I study molecules called polyamines, and I'm going to tell you a little bit about why. So this is a pretty busy slide, but this right here is a cell, and all of these different functions the polyamines do. So they're involved in increasing protein clearance, they regulate cell growth, they're involved in neuronal stability, and they regulate neuronal activity. So what are polyamines? Polyamines, you get them from your diet, you get them from plants, you get them from fruits, but our body also synthesizes them, okay? And it goes through this process that makes these small little molecules, these flat small little molecules that are called polyamines. And what is poly? Poly means many. Amines are these amine groups right here at the end. And so our body synthesizes them and makes bigger and bigger. So you have 3 amines here, 4 amines here, and the unique thing about polyamines is that in our body they're physiologically charged, okay? So they have positive charges. And since they're unique, it's flat, they like to interact with a lot of different things, structures, receptors, proteins, microtubules, tau. And so our body has a very good way of regulating these levels of polyamines. So you have the production to go from these lower order species to the higher order species. And then the body is very good at recycling them. So they recycle back. And during that recycling process, it converts it to an inactive form. And that inactive form can either be removed from the cell, or if the body needs it, it'll be recycled back. All right? What we find in tauopathy models or preclinical models of tau is that there's an excess of these inactive products, all right? And so there's a reduction of the ones that are doing all the physiology that we need, but there's an over excess of these particular products. So I look at trying to understand why this is happening, and maybe we can come up with strategies to either decrease the amount of inactive products and boost the amount of the active products. So there's 2 different assays or techniques that we use to measure the amount of tau clumping or the tau aggregation, and we call that a ThioT assay. And basically that is this line up here, or over here. If you look at the black line right here, the height of this line indicates the amount of tau that is actually aggregating, the clumping that we call. Another assay that we use to measure tau's function, the microtubules, which are right here, they form these microtubules, and again, that supports the structure. We're able to measure the polymerization of this assay too, and this is in this graph right here. When we add polyamines to the system, if we look at this black line here, this is the amount of tau that clumps or aggregates over time, right? If we add the active polyamines to it, we actually decrease that clumping. All right? But if we add the inactive products to it, we no longer have that effect, and actually it starts promoting more tau clumping. All right? If we look at microtubule polymerization, and remember that's tau's function is to stabilize the microtubules. As this, there's several processes that go on with the microtubule polymerization. This is tau is in the black line, and as you add in polyamines, you actually shift this little curve to the left. And what that means is that the polyamines are stabilizing the tubulin and making them polymerize even more. All right? But when we had these inactive products, you remove that effect. So the polyamines are actually doing two parts. It's inhibiting the tau from clumping, and it's also polymerizing the microtubules and supporting tau. And so one of the treatments or one of the therapies that our lab does is in a lab is called gene therapy. And so we can make AAV or viral proteins and overexpress the enzymes that promote the production of these. All right. And so one experiment that I'm going to share with you that we just got is this particular enzyme called spermine synthase, and this boosts and increases the higher order polyamines in the cell. So we inject this right into the brain where so these are tau transgenic mice, and one behavioral outcome that these mice experience with age is that they start to become memory impaired. And we can test their memory by putting them in this little swimming pool, all right? And we call this a radial arm water maze. So this has 6 arms, and there's a little platform there. And we stick different goal arms right here, and then we'll put little cues on the wall like a Biogen sign or an exit sign. And we'll stick the mice in one of these arms, and they'll swim out to the middle, and they'll look around, and they'll find the platform, all right? And so we'll do this 15 times, all right? We'll put them in a different arm every time, and they'll swim out, and they'll make an error. And so on this axis right here are the number of errors. So you can see the tau mice are in blue, or the tau mice are in the red, and our gene therapy product that boosts polyamines are in the blue. And they both start out making an average of 5 to 7 errors, all right? And we keep testing them 15 trials, and they actually learn. They learn the task pretty well, all right, both of them. And we'll come in 24 hours later, we'll do it again. And so you can see they start out low, they remember this, all right? And then we play a trick on them and switch the task 180 degrees. All right, and so now they need to reorient themselves. They need to relearn the task again. And so you can see once we do that, they don't like it, and they make like 12 errors up there. All right, and so again, if you look at the red line, this is the tau mice. They learn much slower. But the mice, when we're boosting polyamines, they actually learn quicker, and this is actually pretty significant. So this kind of leads us to think that we need to kind of boost the polyamine levels within these tau transgenic mice. Another technique that we use is called in vivo microdialysis. And so this is a technique that we can implant a probe into the brain, and it'll sample the environment of the brain. And so we're able to see neurons, glia, blood vessels, and these molecules, tau, polyamines, will pass through this little membrane, and we're able to infuse a solution in and infuse it out and collect it. We're able to do this over the course of a couple of days and get real-time measurements of how tau is changing or how polyamines are changing. And this is pretty much the setup of how it is. Mice sit in this little cage, and it moves. It has a sensor. And we call this they have a little crown there that, and again, this is just another depiction showing that. This is my my postdoc who sets this method up, and I can't do this. She does all the work. And again, you see these little molecules that can pass through, and we collect it. And basically, okay, well, there's some they actually move. You can see it moving. But in long story short, what we can see is that in our non-transgenic mice that don't have tau, we can measure the inactive products here, all right? And we don't see any major change of these inactive products, right? But if we infuse a solution that increases the neuronal activity of the mouse, what we see is this big spike in the inactive products, the inactive polyamines. And what that's saying is that they're being held in the neurons or some type of cell, and during activity they're being released. And that doesn't seem to be a good thing. So in our tau models, in our tauopathy models, this particular pathway we may be able to target. So this is just kind of a summary showing that during tau aggregation in the polyamine hypothesis, all right? Tau, again, normally stabilizes the microtubules. The microtubules are free, and it causes polymerization and stabilization of the microtubules. But there's some event, either a mutation in tau or some aging, something happens in where tau can fall off the microtubules, and tau can start to aggregate and form these clumps. What we find is that polyamines, they can act as these endogenous inhibitors of the tau aggregation. And they can also stabilize the tubulin. All right, that's my time. I'm going to go ahead and wrap up here. So again, they serve as small bioactive molecules, and these might be used as good therapeutics for future targets. Thanks.